So our final speaker for the panel here is Dr. Luis Tedeschi. Luis is a professor of animal nutrition at Texas A&M University. He's a Texas A&M Life Research Fellow, AgriLife Research Fellow, a Chancellor EDGES Fellow, and an Honorary Professor at the University of Sassari in Italy. He serves on the modeling committee of the National Animal Nutrition Program, where he oversees modeling efforts related to livestock production. He earned his Ph.D. in animal science, once again, another Cornell grad. I think we have a theme going on here, and has published, well, we know he's published hundreds of papers. So welcome, Luis.
Thank you, Karen. So Karen told me I only had 5 minutes or even less, up to 5 minutes, so I had to delete most of my slides, and I only kept 100 of those. And I'm not even sure if the slide is up there. Yeah, there we go. So, first of all, thank you so much for the invitation to be here and be part of this discussion group. It was really nice to actually be here during the morning to see all the discussion of the other groups. As a nutritionist, and I think most of the nutritionists here in this panel, I think we understand all the previous discussion asking about how we measure methane. So we are also asking how do we measure methane. So even the nutritionists are not even so sure how we do it. So I only have 5 points and 100 slides to go. So I'm going to start with these points. First of all, precision versus representation of measurement tools. We have high accurate tools. I think Karen was talking about that, like respiration chambers in direct calorimetry. But they're limited in scale and ecological realism, or depending where we're using those. Conversely, we have sniffers and tracer gas methods that gives us a wider reach, but introduce some uncertainty. So from 2 papers that we published, one in 22 and one in 23, we went over and we discussed most of these methods that Karen presented in her introduction. So respiration chambers are accurate but not field representative. That's a given. Green feed and others, they're very good compromise, but requires repeated visits and controlled conditions. I think Sarah brought that up. And we don't have control over that. She also mentioned that. SF6 as a tracer or any other tracer, suitable for grazing animals, but with its own greenhouse impact and variability of the method. In terms of methodological variation, we have problems in terms of meta-analysis. So in our 2023 paper, we mentioned some works that they analyzed close to 400 studies, and they found very inconsistent methods, or data that were showing very high inconsistency between methods, reporting different standards and gas recovery rates that actually impact the prediction, not within the experiment, the study that they were conducted on, but also hampers the meta-analysis. So we have to remind the meta-analysis, we want those studies to be as similar as possible. The third point is the in vitro and in vivo. We still need some sort of a mechanistic modeling or mechanistic approach to bridge these methods. We often test additives, feed additives, or any other additive, or feed, or diets in vitro and then scale to animal systems. But the translation isn't always linear, if that is any linear relationship. So therefore, mechanistic modeling should be something we need to discuss to bridge this limitation of in vitro and in situ. We may need to change paradigms here. Top-down and bottom-up approaches, we discussed this in our papers from the 22 and also 23. So bottom-up methods, they're really focused on the cow, and that's most what we do in terms of modeling, nutrition modeling, I should say, whereas the top-down is really focused on whatever is up there in the sky, and then try to associate or allocate where that methane is coming from. And we know that this reconciliation is not always straight. And there are several papers out there talking about these discrepancies. My last point, and then I can go to my 100 slides, the marginal gains of cattle-specific mitigations, even with 100% reduction in cattle methane emissions, 100% reduction in all of the emissions, at least data from EPA says that this represents 5 to 6 to 7% of the total CO2 equivalent in this country. And I do understand that this varies. It changes from one country to another depending how that data is collected and how that data is analyzed. The point being here is this. We have to be smart in how delivering this idea of a reduction of methane. Perhaps we need to combine with other ideas, not only reduction in methane. And it is site-specific strategy. A blanket solution is not going to work, and I think most of the speakers in the morning alluded to that. And it has to be site-specific, and it has to be data-specific for those locations too, because it depends on not only the animals that we're analyzing, different species, same species, but also what they're eating and the management of these animals.